It's Monday, August 2nd, 1976, and a representative of Pennsylvania's branch of the American Legion has just made a terrible announcement. 4 individuals of its chapter have died. They'd all just returned from a convention that they attended in Philadelphia, and they began complaining of a flu-like illness. What was this disease? Could it be the very much feared return of the 1918 Spanish flu? It appeared that a soldier in Fort Dix in January of 1976 had fallen ill, and it appeared that was an influenza virus that was similar to that of the 1918 flu. This illness was one that did have the symptoms of influenza in that there was fever, there was body ache, but there were also other characteristics that were not consistent with influenza. For example, family members of the Conventioneers did not get ill, so it did not seem like a contagious illness. By August 4th, 1976, the CDC had already determined that these diseases were not caused by the influenza virus, nor were these diseases caused by other usual suspects, such as the bacterium Streptococcus pneumoniae. But by the end of the summer, almost 200 patients had been hospitalized, and there had been up to 29 fatalities. The CDC had a mystery on their hands. They had to figure out 2 things. How did this agent, whatever it was, get into these patients? And 2, what was the agent? The Legionnaires that attended the conference in the Bellevue-Stratford Hotel in Philadelphia, 6% had ultimately fallen ill. So it was suspected that maybe there was something presented at the conference, food, something like that, may have gotten them ill. But there were already observations that were bringing that hypothesis into doubt. There were individuals that had not attended the conference that had fallen ill, but they had walked down Broad Street right in front of the hotel. So soon, the CDC investigators began to question whether there was something in the air being emanated from the Broad Street Hotel. Quickly, they looked at the air conditioning system, and the air conditioning system was an old industrial style with a large cooling tower on the surface. It was somewhat faulty and spraying water onto the street below. The air conditioning system also had a vent in the lobby, which is a location where investigators believed many of the people had contracted the disease. But the CDC investigators thoroughly checked out the cooling system, and they could find no organisms that could have caused the disease. This was a mystery. They had a good idea of how these individuals got sick through what mechanism, but they had no idea what the organism was, and they wouldn't know that organism for another several months. The CDC investigators were getting desperate, and ultimately they started looking at organisms that were unlikely to cause this type of a disease. They had to be sure it wasn't an unusual presentation of some disease. Among other diseases, one that was investigated was Q fever. Joseph McDade, who is an expert in rickettsial diseases, was asked to rule out Q fever as one of the diseases. So he went into patient tissues and he started looking for the organisms Coxiella burnetii that would cause Q fever. And like everything else, he couldn't find it. McDade had been given one task, to determine if this disease was indeed caused by Coxiella burnetii, the organism that causes Q fever. And he had done the tests exactly as is described, and he ruled out Coxiella burnetii. The disease was not Q fever. But he did see some isolated rods in the tissues, and he investigated them using gram stains, but they didn't show up. So McDade ultimately reported back to supervisors that it wasn't Q fever that was causing illnesses in these individuals, and he was released back to his regular work studying rickettsia. But he had never forgotten about this disease and the strange little isolated rods that he saw in the lung tissue of these people that had passed away from this mysterious Philadelphia pneumonia. So, over his Christmas break, he takes it upon himself to pull the samples of the diseased lung, and he starts looking again. He performs the staining again, and he looks and looks and looks, and ultimately, he finds something that's interesting. Not a single rod, but a cluster of rods, suggesting that this rod was indeed something living. It might be bacteria. So he decides, I'm not going to run this test to look for Q fever. I'm going to run the next test to look for any bacteria. So he excludes a reagent that is critical to the Q fever assay, an antibiotic. With the exclusion of this antibiotic, now other bacteria that might be in the lung tissue can grow. So he takes the lung tissue from these diseased patients, he puts it into the body cavity of guinea pigs, and then he takes a guinea pig spleen material and he applies it to embryonated eggs, chicken eggs. And what he finds ultimately is that bacteria begin to grow. He then takes serum from the patients that had died or been ill of this disease, and he knows that these might contain antibodies, and antibodies should be able to latch on to whatever was the cause of this disease. So he performs this test, and antibodies from these patients do indeed bind to some bacterium. So he takes blood from individuals that didn't have the disease, they lacked the bacteria. He then does another test. He takes the blood samples from the people that had suffered the disease, he takes samples that were isolated very early on, and then he takes samples that have been taken later on in the course of disease. And what he finds is the emergence of this antibody. So it really appears that this bacterium that these antibodies were picking up was something that had been acquired approximately at the time of the illness. So this ultimately leads them to identify a brand new bacterium. The disease at this point had been called Legionnaires Disease. The bacterium that was ultimately shown to cause this disease became called Legionella pneumophila. Now, this organism, Legionella pneumophila, is an interesting organism. This is a bacterium that lives inside of other cells, and its usual host are things like amoeba, protists. These are organisms that live in stagnant waters, and the bacterium, Legionella pneumophila, enter into these organisms and live their lives there. And that's why this organism is associated with things like cooling towers, not well-kept hot tubs, any stagnant water. So the organisms live in these amoeba, and then when that water becomes aerosolized, it can be inhaled by individuals, and the bacterium goes into the lungs. And in the lungs, it finds a type of cell that is roaming around. It's a defense cell, and it's trying to find bacteria to rid from the lungs. And this cell, this pulmonary macrophage, does indeed engulf these bacteria, but the bacteria have the nasty trick of being able to survive inside of these macrophages. So ultimately, how did this outbreak come to an end? Well, it seems just by coincidence, the hotel employees changed the filter in the air conditioning unit that was spraying air into the lobby of the hotel. And with that change, it reduced the number of airborne bacteria such that fewer people, if any, were getting infected. But this disease, Legionnaires' disease, still continues because of poorly maintained hot tubs and things like that. So it's important to make sure that all these devices that use water are properly maintained so that the amoebas don't grow in the water and thus the Legionella, the bacterium, don't grow in them.